Plant growth and development is a continuous process that begins from seed germination and progresses through seedling emergence, leaf formation, height expansion, and biomass accumulation. Each stage reflects important physiological changes that allow the plants to establish, photosynthesize, and adapt to its environment.
The objective of the practical are to observe and analyze plant growth over a 35-day period, focusing on early developmental stages, then to monitor germination, seedling emergence, leaf production and the appearance of axillary buds, and to measure plant height at 5-day intervals to track vegetative growth patterns. Next, to record fresh and dry biomass at week 2 and week 5 to assess changes in plant weight and overall growth, and to capture close-up morphological footage to visually document structural development. Lastly, to integrate visual observations with growth data for a clearer understanding of how the plant progresses from seed to seedling under standard crop management conditions.
The apparatus that we use is bottle, cutter, marker, seeds, scoop, and soldering iron.
Next, for preparation, we prepared the setup by cutting the bottles, making drainage holes, and tagging each plant for easy identification.
In this section, we explain the crop management practice used throughout our experiment. These practices cover the planting medium, watering schedule, fertilizer application, and the location of the experiment. Each step is carefully planned to ensure healthy growth and accurate result. For media preparation, we use a mix of topsoil and cocopeat in a 2 to 0.5 ratio. Topsoil provides essential nutrients while cocopeat improves aeration and support healthy root growth. For watering, we irrigate daily during germination to soften the seed coat and trigger growth, but we avoid watering on rainy days to prevent root growth. For seed planting, we place the seeds evenly in the medium and cover them lightly with soil to ensure proper germination. Fertilizer is applied when plants develop 3 to 4 leaves and then every 5 days. This timing ensures the plant receives enough nutrients during rapid vegetative growth. Our experiment is placed in an open area with direct sunlight to maximize photosynthesis and ensure the plants are not shaded.
Germination is the process where a seed develops into a seedling. The radicle or first root emerges from the seed coat, breaking it, and grows downward into the soil to absorb water and nutrients. Seeds don't need light to germinate as they rely on food stored in the endosperm. In dicotyledons, germination produces a root and 2 cotyledons to support early growth.
Seedling emergence occurs when the cotyledon or coleoptile becomes visible above the soil surface. In dicotyledon plants, germination produces a seedling root along with 2 cotyledons. These cotyledons also contain food reserves that support early growth of the seedling.
Leaf production is observed when the petiole becomes visible above the soil. After seedling emergence, the apical meristem of the terminal bud generates the first true leaf, followed by additional leaves developing alternately along the primary stem to enhance photosynthesis and vegetative growth.
Now, let's look at the result of our experiment and discuss the observation.
The height of mung bean plant increases gradually from 4.33 cm to 9.49 cm during the initial weeks. After third week, the plant exhibits grow rapidly as the young leaves begin to undergo photosynthesis. This process produces more energy, promoting cell elongation, cell differentiation, and cell division, which contributes to the rapid increase in plant height.
The average number of leaves increased gradually from 1.63 to 2.75 for the first 2 weeks, then rapidly grow after it. The growth successfully demonstrates mung bean plant have absorbed water and nutrients via the root system. The mature young leaf also undergoes photosynthesizing efficiently to produce necessary food for growth.
The average of fresh weight of mung bean plant is from 1.59 gram to 12.24 gram. The graph shows that the plant develops more leaf for enhancing light interception and maximizing photosynthesis. The larger root system improves water and nutrient absorption. This increases the rate of biomass accumulation and water uptake, leading to observed rise in fresh weight.
The average dry weight increased from 0.33 gram at week 2 to 2.46 gram at week 5 because the mung bean plants were growing and producing more biomass over time. As time passed, the mung bean plants undergoes active cell division and photosynthesis, which produce sugars that were assimilated and stored in plant tissues such as sperms, thus increase the dry weight.
Overall, the mung bean showed slow early growth followed by rapid vegetative expansion after week 3. The early height increase reflects seedling establishment, while the sharp rise later is driven by active photosynthesis, supporting cell elongation and development. Fresh weight increased from 1.59 gram to 12.24 gram, and dry weight rose from 0.33 gram to 2.46 gram, showing strong biomass accumulation as small leaf and roots form. These results highlight that mung beans grow quickly once their photosynthetic area and root system are fully developed.